Good afternoon, colleagues. Maybe there's learners also watching the video. I hope that we can help you today with some of exam tips and quick wins. We accumulated a lot of marks. So if we look at grade 12, paper 2, the content is the code of life, meiosis, genetics, and then the one of evolution. But let us look at how can we break it down for you and give some crucial information and tips on these topics. The first thing that is very important when we look at the code of life is DNA replication versus transcription. So sometimes straightforward answer in the exams, but the way we are going to answer it, that is a problem when we start marking. So I highlighted the DNA double helix molecule unwinds. We can not only state the molecule unwinds or the DNA unwinds, it must be the DNA double helix unwinds. Also, we must say each strand serves as a template. And not just the DNA serves as a template, you must say each strand. And then we must indicate that it's 3 DNA nucleotides that will form a complementary base. So the difference between DNA replication and transcription also very important. So always one of these will be asked in the exam. So the one is the double DNA helix unwinds, and one strand is used, or each strand is served as a template. In this case, in transcription, it is one strand. In DNA, it's DNA nucleotides that we are using to form a DNA replica, and in transcription, we use the RNA molecule to do that. And in DNA, the product is 2 genetically identical DNA molecules, and in RNA, the product is a mRNA complementary to the DNA, and that is the product. So maybe they can ask it to explain, or they can ask the difference. So emphasize on how to explain. We need those highlight marks. In translation, it is very important to look in translation. The anticodon of the tRNA matches the codon on the mRNA, but if they ask you a specific example, then you must say the anticodon of the tRNA, and maybe you have to mention the anticodon. Maybe it is GCC, will matches the codon on the mRNA, and that will be CTT. So look at what they ask and bring the required amino acid. Maybe they refer to a specific acid, then you must name. So now you must take your knowledge on translation and you must be able to apply it on a specific example. Code of life, it's very important to know your diagrams. So you have to identify that V is the mRNA, U is the anticodon, T is the tRNA, S is the amino acid there. And a lot of amino acids that binds together is called a polypeptide. And the ribosome, you must be able to identify the ribosome, and you must be able to apply your knowledge that in the ribosome the process of translation is taking place. Also, the binding between 2 amino acids, and that binding is called a peptide binding. Then they ask always something like give a table, and they indicate the amino acids. You don't have to know the amino acids, but in this case, the table below represents DNA. So therefore, the red around DNA and the DNA base triplet. This is the DNA base triplet. Sometimes they will say this is mRNA. Then they will ask it, you must determine the DNA. So look what is the table. What is the heading of the table? DNA or RNA or mRNA. So that is very important. So give the DNA base triplet of his. So you will go to his, and then the base triplet is GTG. Or they will ask the codon of his. Then they refer to the mRNA of his. So if we go to his, then we will see GTG is the DNA, so the codon will be CAC. So be careful, read carefully, read very carefully on what they ask, and also they can ask, give the nitrogen base of G, for example, that is guanine, or T, that is thymine, so also know your different nitrogen bases and the letters. Right, so if I look at DNA and RNA, that is the differences between DNA and RNA. Again, you have to read very carefully. If they ask what is the differences between DNA and RNA, you can mention any of these. But if they ask what is the differences in the nucleotide base, or what is the differences in nucleotides of DNA, then we only can refer to the nitrogen base is thymine, and the nitrogen base is uracil, and contains deoxyriboses, and the and RNA contains riboses. So read carefully, what do they ask? Do they ask the differences, or do they ask the differences in nitrogen bases? A typical question again, they can give you a sequence of a protein and a sequence of amino acids that is binding there. And now they can ask, for example, what is number 1? What is the DNA base triplet of valine or val? So I go to val and I can see on my table, I can read it off CAC. So just read carefully, what do they ask? Do they ask the base triplets, or do they ask the mRNA or the tRNA? Do they ask amino acid number 3, or do they ask amino acid number 7? All right. So in this case, we see that there is important mutation that did take place at number 6. And now usually they will ask, explain the mutation. What happens now? So first of all, you must indicate where the mutation is did occur. So what is the change in the sequence of the DNA base triplet? So that is a mutation, and specifically a gene mutation. So you will indicate the position number 6, and then you will indicate that glutamine or GLU was replaced with VAL, and you explain how this mutation did take place. So the CAS on the val changed to GAG on the mRNA, and therefore a new amino acid was formed. So specifically refer to the different codons and anticodons that was formed when they ask, explain the mutation. Sometimes there is a mutation, but then the mutation, the new codon or anticodon will also code for the same amino acid. So there is a mutation, but there's no change in the amino acid, so there's no change in the protein. So be very careful, look and read very carefully if the mutation does bring a new amino acid, or does it change the protein. But we must explain mutation according to the DNA that changed, and then the mRNA that changed, and then the tRNA that changed. All right. Something that is very, very, very important is the DNA profiling and how to determine the father. Now, in this case, remember, any baby must have either a DNA profile band from the mother or a DNA profile band from a father. So the DNA profile of a baby or a child must compare with either the mother or either a father. So how do we do this? The first step what we do is we compare the baby and the mother, because we know who is the mother, né? Because she gives birth. When we determine who is the mother's DNA profile bands, we eliminate them. Then the rest of the bands must compare with one. All the bands must compare with one of the males. So in this case, we can definitely see, as on the screen, that male 2 is definitely the father. So, who is the father? Male 2. Now they want you to explain your answer, and then the explanation must be the remaining bands that does not match with the mother. All of them matches with the father, or the male 2, and therefore male 2 is the father. Another thing that I want to emphasize here, I'm not saying that the baby's DNA matches the father's DNA. I'm saying, or my statement will be, the baby's DNA profile bands matches the DNA profile bands of the male. So we are in a hurry, and then you write the DNA of the baby matches, most of the DNA of the baby matches most of the DNA of the father. And that is totally wrong. It's not the DNA that matches, it's the DNA profile bands that matches. So the DNA profile bands of the baby matches the DNA profile bands of the father. And if it's about suspects and DNA that was found in a glass or something, then we will say the DNA profile bands of the glass, or which was found on the glass, matches the DNA profile bands of suspect 1. So we refer to this as DNA profile bands. Make sure that you do that, otherwise it's not correct. So in this case, we don't say more DNA profile band matches male 2, we must specifically mention that the remaining DNA profile bands matches male 2. Meiosis, please look at the different stages, and you must be able to identify these stages, and also you must explain what is the behavior of the chromosomes during these phases. So I just want to emphasize one thing. In metaphase 1, we refer to homologous chromosomes on the metaphase, or on the equator. So in metaphase 1, it is homologous chromosomes. And in anaphase, it is chromosome pairs that separated, or chromatids that separated. So in metaphase 1, it's homologous chromosomes. In metaphase 2, it's only chromosomes that are on the equator, like this one. In this case, there's only chromosomes on the equator. It's not homologous chromosomes, so therefore this will be metaphase 2. Furthermore, you must be able to identify the centriole, the spindles, the centromere, and the phase that is represented here. And also, you must be able to explain the function of the centriole and the spindle fibers and the centromere in metaphase. Then in metaphase, as I said, in meiosis, you must be able to identify the different phases, if I can quickly show you there. So in metaphase 1, we have homologous chromosomes that are on the equator, but in this case we have only chromosomes that are on the equator. So that is a very important word that we must use, homologous. In this picture, the first picture, a chromosome is separated to the centriole, and in this case a chromatid. So this is anaphase 2, but where we have a whole chromosome, it's anaphase 1. So be very careful to look at that in your explanation. We must not forget about non-disjunction, where on chromosome pair 21, homologous chromosomes fail to separate. So I mentioned the pair that doesn't separate, and I mentioned that it's homologous chromosomes that doesn't separate. Chromosomes, sister chromatids, daughter chromosomes. So this is a chromosome. It's bound together by a centromere, which binds two chromatids, and we call this chromatids sister chromatids. So, and in anaphase, we start to form a daughter chromosome. So this is not a chromatid. After they separate, they it forms a daughter chromosome. So be careful when you are talking of a chromatid and when you are talking to a daughter chromosome. Be very careful for that. Please pay attention to the differences between meiosis and mitosis. Remember, meiosis introducing genetic variation, and mitosis is just an exact copy of the chromosomes. So the differences between meiosis and mitosis: meiosis is 2-cell division, mitosis is 1-cell division, etc. So please look at your differences. Long time ago that they asked a question on tabulate the differences or mention the differences. Also in the differences of meiosis 1 and meiosis 2. Make sure that you know what is the differences between them in each phase. The prophase, remember in meiosis 2 there is no crossing over. And in meiosis 1 it's homologous pairs. And in meiosis 2 it's only chromosome, as I mentioned before. Then the importance of meiosis: it introduced genetic variation, and it also have a halving effect of meiosis to overcome the doubling effect of fertilization. So there's 3 very important for meiosis. What is the importance of meiosis? Also a long time they didn't ask that, so therefore I want us to emphasize on the importance of meiosis. Something that I just want to look at is crossing over, the state, how you were going to state it or how you are going to write it. Remember, it occurs during prophase 1, that is important. Then we have to say homologous chromosomes. We cannot say chromosomes exchange genetic material. Sometimes you say it's prophase 1, chromosomes exchange genetic material, and you leave out the fine detail of the process. So it's homologous chromosomes, and when they are lying together, it is usually the non-sister chromatids that overlap. Not chromatids, not any chromatid overlap during crossing over. It is the non-sister chromatids of a homologous chromosome pair, or the adjacent chromatids overlap. They exchange genetic material, and then, very important, the product of crossing over is a combination of genetic material from both parents or 2 genetically different chromosomes. In this case 2, but actually genetically different chromosomes is the end product of prophase. So in random arrangement, we have to say the homologous chromosomes arranged randomly on the equator during metaphase 1, and the single chromosome arranged randomly on the equator during metaphase 2. So again, in metaphase 1 we have homologous chromosomes and not chromosomes alone. So there's a big difference between the homologous chromosomes and chromosomes of metaphase 1 and 2, and you must emphasize on that one and make a differentiation between metaphase 1 and 2. So that is contribution to variation, which is very important. Also, when I look at mutations, you must be able to differentiate between a gene mutation and a chromosomal mutation. So a gene mutation is a change in the sequence of the nitrogen bases of the DNA. So that whole sentence will allocate 2 marks or 0 marks. Chromosome mutation is the number and the structure of the chromosomes. Random fertilization, where any sperm can fertilize any egg. Random mating, any male can mate with any female. So that is very, very important to look at, and the way we answer it, sometimes you know what is the answer, but there's a difficulty in expressing yourself when you are answering this questions. If we look at genetics, genetics is very important that you read. Genetics is like mathematics. There's a formula that you must use and apply when you are doing genetics. So let's focus just on a few points where sometimes you struggle to identify what the answer is. Now, there's a relationship between the chromosomes, the DNA, the gene, and the genome. So the gene is a small portion of the DNA that codes for a characteristic. So there's a gene that codes for a characteristic. DNA, on the other hand, codes for all your characteristics. I'm going to say that again. A gene is only coded for one characteristic, and DNA codes for all your characteristics. So that's the difference between a gene and the DNA molecule. Then the other one that is problematic is a genome versus a karyotype. A genome is the mapping of all your genes on all your chromosomes. That is the genome. And a karyotype is the number and shape, the arrangement of the number and shape of all your chromosomes. So listen carefully, a genome is all your genes on all your chromosomes, and that is the human genome. But a karyotype refers to the number and shape of your chromatids. The number and shape of your chromosomes, and in this case that is called a karyotype, and we can make a lot of conclusions with a karyotype. We will come to that. So very important, the difference between a genome, all your genes. If you have that word in your, give one word the biological term, then you must know all your genes is a genome. Then do not forget about multiple alleles. Remember now, multiple alleles is two alternative forms of a gene at the same locus. So there's two alternative forms. We know that the alternative form is an allele. So in multiple alleles, it means there are two or more. No, I said it wrong there. So there are more than two, not two and more. There are more than two alleles at a specific locus. In this case, with blood groups, we have three alleles. We have the allele capital letter A, we have the allele capital letter B, and we have the allele small letter I. So that is your three alleles. And these three alleles can code for four different phenotypes. Therefore, we have blood group AB, blood group AB, that is codominance, and blood group O, that is complete dominance. So remember now, multiple alleles are more than two alternative forms of a gene at the same locus. And the example that you have to know is blood groups. So they can ask you, explain multiple alleles using blood groups, and then you can say these three alleles that goes for four phenotypes of blood groups. Also in blood groups, I can ask you to explain Mendel's law of dominance using blood groups, and then you can explain complete dominance, you can explain codominance, and also you can explain that the recessive allele is only expressed if it is in the genotype homozygous. And then we can use blood group O for that, to explain that. So blood groups, very, very, very important. The way we are writing blood groups, the alleles of blood groups, and how blood groups express dominance, also codominance, and also how blood groups is a good example of multiple alleles. As I stated before, we look at the karyotype. It is the number and shape of homologous chromosomes on that is displayed, and we can see in this case we have homologous chromosome pair. There it is. We can see that this one autosomes is from number 1 to 22, and then the gonosome is number 23. And I just want to emphasize something on the gonosome. When we refer to this we say this is an example of a male, and the reason why it's not, it have an X and a Y chromosome. We have to specifically refers to the gonosome or chromosome pair 23 that have a X chromosome and a Y chromosome. It doesn't have an X and a Y. We are not busy with math. It has an X and a Y chromosome on chromosome pair 23 or on the gonosome. So sometimes we will ask what is the gender of this karyotype, and then they will say male, and the reason is they have an X and Y chromosome. So we must just be specific. Where is that X and Y chromosome? Always, and don't write just X and Y. So very important there. Always important if this is this a normal human karyotype or is it Down syndrome? How many chromosomes in the gamete? How many chromosomes in a body cell? So make sure that you know the difference between gametes, and gametes is haploid, and body cells are diploid, or somatic cells are diploid. So make sure that you know how many gametes, how many chromosomes will there be in a gamete, and how will this person's gamete, for example, differ in chromosome number. Very important. Then people, if we look at monohybrid crossings, the only thing at this stage, monohybrid crossings is answer very good. We must look at where we allocate the marks. The marks are allocated in that different stages. And if we look at these different stages, we will see we must indicate P1 and F1 on the right position. P1 is on top, F1 is on the bottom. So in this case, we have tongue rollers and non-tongue rollers. Sometimes they use flowers. We cannot say pink and, or for example, red crosses with white. We must say a red flower crosses with a white flower. That is very important when we use the dihybrid crossing. So look at the allocation of marks, and then the position of meiosis and fertilization, also very important. They must be on the right position for one mark to be allocated in that situation. So then I just want to emphasize something on dihybrid crossings. Dihybrid crossing is where we have two genes that are crossed. We have, in this case, a gene that is plant height and a gene that is stem color. And these two genes have alleles. So for plant height, we have a tall plant and a short plant. In stem color, we have a black and a red. So listen very careful what I'm asking. So if I ask what is the two genes that we are crossing here, it will be stem color and plant height. If I ask you what is the dominant phenotype, the dominant phenotype for plant height, so the dominant phenotype for plant height is tall. What is a dominant allele for plant height? It's only the letter T. So read very careful. If I ask phenotype, you give the phenotype, the tall or the black or the red. If I ask allele, then you must look at the capital letter T or capital letter B or small letter b. So what is the recessive phenotype for stem color? It will be red. But what is the recessive allele for stem color? It will be small letter b. So read very carefully what they are asking in a dihybrid crossing. You don't have to be able to do a dihybrid crossing, but I can give you a dihybrid crossing like in this example, and I can ask you to write down the gametes of 1 or write down the genotype of 2, etc. So read carefully what they ask in a dihybrid crossing. The types of dominance is very, very important in genetics. Either you have to give the biological term, or maybe we give you the biological term, we say it is codominance, and now you have to explain it. So also in this case you must make the definition part of your example. So the definition of complete dominance is one allele is dominant and the other one is recessive in such a way that the recessive allele is masked by the dominant allele, and then you have to add in the heterozygous position, because that is the new definition in our exam guidelines. So let us see, if we ask, maybe they will ask what type of dominance is illustrated in this picture. So we will say complete dominance, explain your answer, and then we will say one allele, the red allele, is dominant over the white allele, or it masks the white allele. Therefore, all the flowers will be red in a heterozygous condition. So when you take the definition and you apply it to the example. The same with codominance. Remember now, both alleles of the gene are equally dominant and both alleles are expressed. So what happens in the exam, maybe you have stage fright or exam jitters or whatever, and then you say both alleles are equally dominant and both are equally expressed in the phenotype. They are not equally expressed in the phenotype. Both alleles are expressed in the phenotype in a heterozygous condition. That means if the genotype is heterozygous, it will be expressed. So both alleles of the gene are equal dominant. So at codominance, they are equal dominant and they are both expressed in a heterozygous condition. But if we look in incomplete dominance, neither one of the two alleles of a gene is dominant. Not one of the two alleles is dominant over the other one. And then, very important, we have an intermediate phenotype in the heterozygous condition. So make sure that you know exactly how the definition is stated and know that you can explain it in an example. So in this one, we will say neither one of the two alleles, red and white, is dominant over one another. It results in a, I think it's pink, intermediate phenotype in the heterozygous condition. So that is very, very important to look at your different dominance, types of dominance. Then, in short, is the Mendel's laws, the law of dominance. I also already said that you must look at the example of blood. Mendel's principle of segregation, make sure that you know what is the definition, and Mendel's principle of independent assortment, and also know what is that. So Mendel's principle of segregation in monohybrid crossings, we will have that no two alleles will be during meiosis go into the same gamete. So alleles will separate. So make just sure, especially your level 7 learners struggle with this when we ask it in the exam. So make sure that you know your definition and that you can apply your definition to an example. In this case, I just want to emphasize something. The way we are writing a dominant allele and a recessive allele, we must use the capital letter and the small letter. We can't write capital letter A and then a capital letter A, but just smaller, and think that is representing the recessive allele. The recessive allele is represented by a small letter. So in this case, it will be wrong if you use capital letter A and just a smaller capital letter A. So the way we are writing the dominant allele and the recessive allele, use the letters that I give you and use it as the way we are writing it, as a capital letter and as a small letter. How sex is determined in humans? Normally this is a straightforward question, but and it counts 6 marks, but when you start marking it, then learners struggle because the way they are answering it. So we will say, learners will write female have an X and an X and male have an X and a Y. If the X fuses with the X, the offspring is a female with XX. So that is 0 marks. Because we must say females have on the gonosomes 2 X chromosomes. And males have on the gonosome an X and a Y chromosome. Please leave out a big chromosome and small chromosome. We're not talking about that anymore. It's an X chromosome and a Y chromosome. So if the X chromosome of the female fuses with the X chromosome of the male, then the offspring will have 2 X chromosomes on the gonosome, which results in a female. So I know you know this, but the way you will express yourself, that is something else. So I just emphasize on X chromosome on the gonosome, and it's not the XX and the XY. So please look at that. Then if I look at pedigree diagrams, pedigree diagrams is something that you must practice a lot. But let us see, if they ask a pedigree diagram on sex link, so they indicate to us Hunter syndrome, the males are the black ones and the unaffected ones is white, and the square is males and the circles is females. They don't have to give us a key, it's not necessary, so they can leave out the key. And then we must know that squares represented males and circles represented female. In this case, we can go and allocate the gene type of each individual, because we can now know that John is Hunter syndrome male, and in the opening statement I will tell you which symbol to use. So if they have Hunter syndrome, then it means it have a recessive allele, because sex links are a recessive allele that is carried on the X chromosome. So it's very important to know your definition of a sex-linked disease. So in this case, it's very easy to allocate the genotypes of the males. So lucky is also not so lucky. He have a recessive allele on the X chromosome, therefore he has Hunter's syndrome. But now it comes to Ethel. So Ethel is unaffected female. So in fact she can have 2 types of genotypes. She can either be capital letter T, small letter t, or she can have capital letter T, capital letter T. But the reason why we allocate the small letter T with Ethel is the Y chromosome from John is carried to Lucky, because Lucky is a boy, so the father gives him the Y chromosome. The Y chromosome of his chromosome goes to Lucky. And the X chromosome, Lucky inherits from its mother. And in this case, because he's Hunter syndrome, he inherited from his mother, so his mother is a carrier, although she's not affected, she's a carrier of this syndrome, and therefore Lucky inherited the X chromosome that carries this Hunter syndrome, inherited from his mother. So therefore we can establish that Ethel is heterozygous for Hunter's syndrome. So she's a carrier. So make sure that you definitely know how to write sex-linked genotypes, if it's a male or a female. And the female will only be affected if she has a homozygous condition. And that brings me at another one. You must go and look at sex-linked disorder. Why females have a smaller chance of suffering from hemophilia or any other sex-linked disorder. And the other one that they ask is, why are there more males that have color blindness or any other sex-linked disorder? So it's all about females that have 2 X chromosomes. So if they have the recessive allele on one of the X chromosomes, they have another X chromosome that can mask the other one. And with males, they only have 1 X chromosome that can carry a recessive allele. So if they have the recessive allele, the chances is 100% that they can have sex-linked disorder. So please look at this. It's popular questions. It is there in the question paper, especially when there are sex-linked disorders. Then brief outline of the process of cloning. You must understand it and you must be able to apply the knowledge. So in short, when we remove a nucleus, usually the nucleus is diploid and we don't want that characteristics. That is why we remove it. When we insert a nucleus, then we need that characteristics and the nucleus is usually diploid. And why doesn't the offspring looks like the surrogate? Because the offspring have the whole set of diploid chromosomes from the donor who donate a diploid nucleus. So make sure that you know your cloning and why do we remove it and why do we insert a mammary cell? Because it's diploid and we need that characteristics. The same with biotechnology. Make sure that how do we use bacteria to produce insulin or produce any other medicine. We use the plasmid. We take the desired gene, we place it in the plasmid. The plasmid is now recombinated. We place the plasmid back into the bacteria. We allow the bacteria cell to reproduce by mitosis because it's a it's a quick process and many bacteria cells can be formed, and then we extract, in this case, the insulin or whatever we need. So make sure of cloning and biotechnology. It's things that we skipped in classes. They are not much evaluated over the past years, especially biotechnology. So go and look on questions how they ask you to use biotechnology. Cloning and biotechnology for genetics is very, very important. The other thing of genetics is when we have to explain the disadvantages of cloning or genetic engineering, either cloning or biotechnology. Look at your disadvantages and your advantages and make sure that you can apply it in a example. So in this case, the disadvantage is expensive. It will be very expensive to buy a fish banana or banana fish or whatever this thing is. It interferes with nature. It's not normal. I don't know if that's normal, and we don't know the health impacts and the long-term effects. It's all there, but apply it on an example. That is very, very important. People, I quickly want to refer to natural selection and the new way we must answer natural selection. And if you did natural selection in class, you will follow it with me. In the past, we stated natural selection. There's variation. We have long necks and short necks. Long necks are favorable characteristics. They survive. Short neck is unfavorable characteristics. They die out. But now the approach is a little bit different and more scientifically correct. If we explain natural selection, then we state the characteristics that varies. In this case, then we will state the first statement will be there's a variation in the length of giraffe necks. Not again, there's a variation in giraffes. So specifically, what is the variation? In this case, the variation is the length of giraffes' necks. Then describe the variation. Then we will say there is giraffes with a long neck, and then there's a giraffe with a short neck. The third one that is also new, now you must state the selective pressure. And which one will be favorable? So when we eat leaves or food was only available on top of the trees or on higher trees, natural selection took place between giraffes with long necks and short necks. So if it was dark-colored and white-colored mouse, then we will say the selective pressure was that the owl in the night can only see the white mouse or was hunting in the night, and therefore there was this natural selection between white mouses and dark mouses. So please, we must state the characteristics that varies, then we name the variation. Then we must say, what is this natural selection? Which force is going to select? And now we state the unfavorable characteristic and why is it unfavorable. We can no longer say short necks was unfavorable, they die. We must explain, we must elaborate and say short necks was unfavorable characteristics. They couldn't eat enough food. They haven't got long necks to eat the leaves on the top, so they die of hunger. And then we state the favorable characteristics. We say the giraffe with the long neck have a favorable characteristic. He could eat more, he could survive. So when he eat more, he survive. And then what happens? We will say that this characteristic of having a long neck survive. These giraffes reproduce, and they reproduce offsprings with the desirable characteristic, which is long necks. So just a fine-tuning on how to answer it. Not anymore, there's a variation, favorable and unfavorable characteristics. Long necks is favorable, they survive. Short necks unfavorable. So we must elaborate in our answers, and that is very, very important, especially with natural selection. So the next one I want to jump to is speciation. The same with speciation, we must apply speciation onto an example, and as it is stated in the exam guidelines, if a population of a single species, now mention the original species in the exact that they refer to. So we will say, if the population of single rabbits or single tortoises are separated, and then we will mention the specific barrier, river, mountain, lake. They won't give a mark if you say become separated by a geographical barrier and you don't mention the barrier. Then we go to the normal. Then we say the population is split into. Mention how many islands or populations they are split into. They are split into, the original populations was split into 3 or 4 populations. There were no gene flow. Since the population may be exposed to different environmental conditions, they will be different, and you mention that there is natural selection independently, and they differ genotypically and phenotypically, and you mention the differences in the extract, and then even if the two populations are mixed together, they cannot reproduce fertile offsprings, and now there are new species are formed, and you specifically mention those species. So in last year paper, let us see this one and how we will apply speciation on it. The present day distribution of 3 closely related species of the dog family, the coyote, jackal and the dingo, is shown in the world map below. Okay, now we will start in a population of a single species of dog family. Now you refer and you use the example in the heading. They become separated by a geographical barrier. In this case, it's continental drift or ocean specific. And they are now split into how many groups? 1, 2, 3. They are split into 3 groups, 3 different populations. And then we will say there are no gene flow between the 3 populations, and the 3 populations undergo natural selection independently, and they could not mix, or even if they are mixed together, they cannot produce fertile offspring. So now there are 3 different species, and you refer to the different species as coyote, jackal, and dingo. But the opening statement is if a population of a single species, the original population, the original population of dog family was separated by a geographical barrier, you name the geographical barrier. And for those 3 sentences, you will receive 1 mark. So it must be stated correctly. That is in speciation very, very important. All right. The last thing that I want to emphasize on is humans versus the African apes, similarities and differences. Look at the similarities and differences, but also in the differences, look at what is the significance of bipedalism, the large brain, the C-shaped spine, the pelvic girdle, which now is short and wide. So in bipedalism, you can walk upright, you can see further, you can spot predators, hands are free to use tools or to protect or whatever. The larger brain with the skull that enlarge, the brain also enlarge, so more information can be interpreted, more coordination, speak, and you can use your hands, or hand coordination is better. Your C-shaped spine that is going to an S-shaped spine is for balance and to distribute weight even, so when you are standing up that you can do that, and also the significance of the pelvic girdle. So that not only the similarities and differences, but also what is the significance, what changes in African apes and humans that make it more significant. Also the teeth, no more canines or no more, they don't use it, they are not large anymore, and they use it now, no more using it for killing prey or predation and predators. They are using it now for soft food, so they don't need large and sharp canines anymore. Also, emphasize on the line of changing. What is the line of changing? So from Australopithecus, Homo habilis, and Homo sapiens, how did the change occur? And from the shift of the foramen magnum to a more forward position, less protruding jaws, and all those features that change the line. And then the last one that I want to emphasize, remember now the evidence of human evolution. We have 3 evolutions. Evidence of evolution, which is fossils, biogeography, modification by descent, your homologous structures, genetics. Evidence of human evolution includes all your fossil records, cultural evidence, and genetic evidence, which is your mitochondrial DNA. And then evidence of out of Africa hypothesis is only fossil records and genetic evidence. And you must be able to explain it if they ask. Explain evidence, name and explain the evidence that we have on human evolution. So from my side, that is what I have for you on tips, exam tips. I hope maybe some of the things are highlighted, especially on the way you answer it. Sometimes you know what to answer, but just the way you express yourself and the way you answer it, there's not enough evidence. Remember, life science is a science, and the science is a specific process that must be followed. And when you are describing protein synthesis, DNA profiling, genetics, it's a process that must be explained in the scientific way. So from my side, good luck for Monday. Good luck for the preparations. Hang in there, guys, and good luck to each and every teacher that is still helping out there this weekend. The learners, good luck to you all. Best of luck. Thank you. Oh, then I must remind you there will be a competition. MTN is hosting a competition. The prize for this question on this is a bucket hat and a t-shirt, something that we can use on your matric vacation. It will come in handy, and you go to FE's Department of Education Facebook, and you can get the question there, and you answer also there. And the question that I'm going to ask you, I even repeat it twice in the presentation. So my question today is, what do we call the arrangement of all your genes on all your chromosomes? The arrangement of all the genes on all your chromosomes. And you can go to Facebook and you can submit your answer for a bucket hat and a t-shirt. Thank you very much.